and welcome to our next video in our new unit on cells. Now remember, cells are part of all living things, and we're going to delve right into what the big hype on cells is all about. So what are cells? Cells are the basic unit of life, the smallest thing considered to be alive, and it is part of our hierarchy of life. Now remember, when we were talking about what the characteristics of life were, we started out with atoms, and then we went to molecules, and then we went to organelles, and then cells. So those things prior to cells are not alive. So this is the hierarchy of living things. So we start out with the cell, and then we go to tissues, finally to organs and organ systems, and then lastly you have your organism. Now it can be as complex as a multicellular organism like a human, or it can be as simple as a protist, which is usually a single-celled organism. Now we have both prokaryotes and eukaryotes. Prokaryotes are a much more primitive form of a cell and were thought to be found a lot earlier. The eukaryotes came along a lot later, and they're much larger than the prokaryotes. What I want you to do now is to pause your video and come up with the differences between the prokaryote and the eukaryote. And I want you to write that in your notes. Do our lists match? Well, why don't you make corrections to our list? I'm going to go through these, and I'm going to look at your corrected list when you get back to class. So, the eukaryote is membrane-bound organelles. It has a true nucleus, more organelles like the ribosomes, Golgi apparatus, rough endoplasmic reticulum, smooth endoplasmic reticulum, and so on. The prokaryotes are found in primitive forms of life like bacteria, has no true nucleus. The nucleoid region is what we call the region where the DNA is. And there's no membrane-bound organelles. Now there is a membrane, but no membrane-bound organelles. It has circular DNA, and it has ribosomes as its only organelle. So if we take a look here, this is a figure of a prokaryote. As you can see in the middle, we have a chromosome in the nucleoid region. There's nothing enclosing that chromosome, that reddish portion in the middle. And those black dots we see all around are the ribosomes. Ribosomes are where we make proteins. Now at the back here you see a prokaryotic flagellum, which is going to help it move. Now if we look here, it does have cytoplasm. The cytoplasm is where all the organelles sit in, and you will find that in the eukaryote as well as the prokaryote. We have a plasma membrane which encapsulates it. We have a cell wall, and we have a capsule or slime layer as well, and we have pili. Pili are going to kind of help it feel, kind of help it move. They have some ways to respond to stimulus, but they are very primitive. And remember, the cell is very, very primitive and very early. So here's a picture of an animal prokaryote. Now, animal and plant, or an animal eukaryote. Animal and plant eukaryotes are different. We have different organelles in them. But let's look at the main structures within a eukaryote. So you will find all of these structures within an animal or a plant eukaryote. So as we can see, we have a nucleus, we have the nuclear envelope, the nucleolus, chromatin, and nuclear pores. You're going to find all of that in your nucleus. That's going to be the same no matter what kind of eukaryote you run into. We have ribosomes again. Remember, what do ribosomes do? If you said they create proteins or they do protein synthesis, you were correct. We find them on the rough endoplasmic reticulum, and we also find them free-floating out in the cytoplasm in eukaryotes. The rough endoplasmic reticulum helps with protein synthesis. We're going to go over a few more of its jobs later. Now we have a plasma membrane. Do you remember when we talked about lipids? If you do, you remember that those phospholipids make up the cell membrane. So we have Golgi bodies, mitochondria. We have all kinds of organelles. We're going to go through exactly what each organelle does. So here's a plant cell. The plant cell, like I said, has a few other organelles. We have chloroplasts, which are going to help us in the process of photosynthesis or creating sugars. We also have a cell wall. That cell wall, if you remember from our organic chemistry unit, it is made up of cellulose. Cellulose is what gives it its structure. It's that hard structure. Remember, we cannot digest it. That's what we call fiber. It also has a large central vacuole. Now, animal cells have vacuoles, but they are very small. This large central vacuole is going to hold and retain water, and it's going to keep this animal or this plant cell very rigid and very firm. Okay, so how big are cells really? Well, you have 50 to 75 trillion cells in your body, and 2 billion are in your heart alone. But I want you to look at this cell infographic. So we're going to go out to this website that I found. It's a really great website that shows us exactly the scale. So as you can see right here, we have a coffee bean, a grain of rice, and a sesame seed, and your Times Regular 12-point font. As we start to zoom in, we see a grain of salt. So look at that coffee bean, and let's just start looking. Now we have an amoeba proteus. That amoeba proteus is a type of protus, and it's a unicellular organism. Next, we have a paramecium, and that paramecium is another protus. Now we're getting into these human cells. And girls, you have human eggs. And look how big that human egg is in comparison to the human sperm cell, and even in comparison to the skin cell. If I zoom in even further, you're going to see a red blood cell. So just think about that in contrast. Now remember, I mentioned the mitochondria. That is one of our organelles within our cells. Look how big that is in comparison to just the E. coli, a prokaryote. So as we get smaller and smaller here, we're seeing all kinds of other cells, and we're seeing cell parts now. So how big are your cells really? They're really tiny. So let's go ahead and move on. So if cells are so small, how do we see them? Well, we have two ways that we actually see them, and we use a light microscope. A light microscope can go up to 1,000 times magnification, and the reason is you have a 10x ocular lens, and a lot of people forget about that. And it can view living things in motion. So we don't always have to have a slide that has been pre-made for the light microscope. And we have these at school. These are microscopes that we will be using in class. Now, we also have the electron microscope. We do not have these at our school. The electron microscope is 1,000 times X, and the cells must be dead and specially prepared for viewing. We have a scanning and a transmission microscope. Oh, let's go back for just a second. And as you can see, take a guess at what these are. Right here, this is from the scanning, and you're going to see the outside of something. This is actually pollen grains, whereas over here, you're actually seeing cells, and you're seeing the inside of cells in the transmission. Okay, so how do cells work? Every cell has a specialized function, so it is shaped a little differently. Right here, we're looking at a nerve cell or a neuron. In a nerve cell, it's shaped this way so it can send messages. It receives messages over at one end, sends it at the other end. So all cells need the same basic structures, and those structures are called organelles. These collectively function to make energy, make protein, move the cell, store and transmit genetic information, destroy, recycle, waste, or dead material, and communicate with other cells. So we have all kinds of things these organelles are doing, and they're working all the time. So the first portion that we have is transmit genetic information in the nucleus or the control center. So the nuclear envelope is the double membrane, the outer and inner membrane that encloses the nucleus. It contains pores to let the genetic information in and out. Well, the DNA always stays in the nucleus. So can you guess what goes out? If you guessed RNA, you would be 100% correct. So that RNA needs to get in and out. It's what's going to send our messages. In the nucleolus, in this very, very dense region, it's actually a ribosome factory. We're not doing anything with DNA or genetic material here. We're making ribosomes. The nucleus houses the DNA, the chromatin, and the chromosomes, and these are probably some new terms for you. The chromatin is the unwound DNA. The chromosomes are the tightly coiled DNA. One interesting fact is red blood cells have no nucleus. So, and actually a better term for those would be red blood corpuscles, because they do not have a nucleus. So when you think about crime scenes and DNA analysis, you're not actually getting your DNA from the red blood cells. You're getting it from the white blood cells that are within your blood. So making proteins, another job we said for the cells and the organelles is making proteins. Well, you have a smooth endoplasmic reticulum and a rough endoplasmic reticulum, and we usually shorten that to SER and RER. The smooth endoplasmic reticulum is called that because it has no ribosomes and it synthesizes lipids. So that's a little different than what the rough ER does. It has it metabolizes the carbohydrates, so it breaks that apart using hydrolysis, and it detoxifies the drugs and poison. So where do you think you're going to have a lot of these? If you said your liver, you would be right. You're going to synthesize a lot of lipids there. This is where everything goes to be detoxified in your body. So anything you eat or drink that could potentially be poisonous has to be detoxified through your liver. Then we have the rough endoplasmic reticulum. The big difference is it has ribosomes, and it actually has different jobs as well. But when we look at it visually, the big difference is going to be the ribosomes there. It makes additional membranes for itself and for the cell. So where we are getting our membranes from is this rough endoplasmic reticulum. It makes proteins destined for secretion. So while our ribosomes are making proteins for all kinds of functions, if they're on the rough endoplasmic reticulum, they're actually making them for proteins that are destined for secretion. This is it for part 1, but we will be doing part 2 soon. Make sure you study up because we will be having a quiz shortly.